Okay, eager students, we just looked at diffusion. Now we're going to look at a special case of diffusion called osmosis. Most people have heard of the term osmosis because it's used in biology pretty frequently, and a lot of people are confused by it, but quite simply, it's just the diffusion of water. So all the rules we learned in diffusion apply to osmosis. Materials are still going to move from an area of high concentration to an area of low concentration. The only difference with osmosis is that material is water. Alright, so let's take our example here. We've got a cell, and on the inside of that cell, we've got a bunch of little sugar particles dissolved in there. Now, outside of the cell, we also see some sugar, but not as much. Now, in these kind of diagrams, when we look at osmosis, we don't actually draw on the water particles because it gets too busy. So we have to think about what's going on. In other words, if we've got a little bit of sugar, everything else must be water, so we got a lot of water. And if we have a lot of sugar, then we only have a little bit of water because that sugar would be displacing that water. So looking at our diagram here, on the outside of our cell, we have a high concentration of water and a low concentration of sugar. Now on the inside of our cell, we are going to see a very high concentration of sugar, and therefore that would mean a lower concentration of water. Now in our example here, the cell membrane is selectively permeable. And normally what we would expect then is for sugar in high concentration to diffuse out of the cell into the area of low concentration down the concentration gradient. But in this case, it's selectively permeable, meaning that some things are going to get through and some things are not. In this case, the sugar is not going to go across the cell membrane. It's going to be blocked by this barrier, but water can. So what we're going to see then, instead of sugar diffusing out, is we're going to see water diffusing in, and that's the process of osmosis. So our high concentration of water is going to diffuse down the concentration gradient into the low concentration of water on the inside of the cell. So ultimately that means this cell is going to swell up because that water is trying to reach equilibrium. Here's another example using some tubing. So we've got some clear glass tubing right here that has a little selectively permeable membrane in it, and we filled both sides equally with water, and we've got a little bit of stuff dissolved on this side and a bunch of stuff dissolved on this side. So what we're seeing here is we're seeing more water on this side, or a higher concentration, than we're seeing on the right side. So the left has a high concentration, the right has a low concentration. So what's going to happen is water from this side is going to diffuse over into this side of the tube to kind of reach an equilibrium. In other words, go from an area of high concentration to an area of low concentration. And this does a really interesting thing, is that osmosis puts pressure on water on this side. It's called osmotic pressure, as that diffusion naturally occurs. And so our level of water, even though it can pass freely back and forth here, is not going to be what we would expect if we just filled up a horseshoe-shaped tube or glass. And so we're going to see two different levels of water. And at that point, it has reached equilibrium. This osmotic pressure is important in biology because it actually helps things like plants move water from their roots up into their shoots.